Hey there, biochemistry students. This is your week 3 and 4 video, and I'll launch right into it. So we left off previously thinking about chemical equations, thinking about reactions, and I highlighted for you what I think is the most important reaction for biochemical systems on our planet. That would be the photosynthesis reaction. I also mentioned that photosynthesis, of course, is not a single-step reaction in reality. It is actually a very multi-step process that takes place in a cellular structure called a chloroplast, which we're going to learn more about in the next units. We're not going to learn every detail of the process. You can always look up images of detailed steps of photosynthesis, and you can find what that looks like. There are two different phases to it, a light reactions phase and a Calvin cycle phase, and it's all really cool. But for now, we're just going to simplify it down to when light energy is harvested by the proper cellular machinery in a plant, the plant can use that energy to combine carbon dioxide and water in such a way as to build a larger molecule called glucose, which essentially is where the energy becomes invested, converting from sunlight energy into chemical energy in the form of glucose. And then oxygen is produced as a byproduct. You can see down at the bottom of this slide some models of the actual molecules involved. There's CO2, H2O, the sun, and then glucose. In this case, the form is pretty clearly an alpha form, down, down, up, down. Go the OHs around the ring from right to left. That would make it glucose of the alpha form. And then oxygen, O2. We did learn how alpha glucose can be combined by dehydration synthesis in order to make a chain of alpha glucoses. Again, we note that the counting around the ring, we count from the top right where there is an oxygen, here not shown in this ring. This would be an oxygen, this would be an oxygen. We number these carbon number 1, carbon number 2, 3, 4, 5, and again, not shown here would be the 6th carbon hanging off of the top left. Okay, and so if we have the alpha form of glucose, the first OH group is pointing down, the 4th OH group is pointing down. There would also, again, not shown here, be a second OH group pointing down and a third OH group pointing up. That's how we draw alpha glucose, down, down, up, down, go the OHs. The first OH group from one alpha glucose, the one that I've drawn on in purple here, is linked to the 4th OH group of a second alpha glucose. 2, 3, you can see that's on the 4th carbon of the ring. And when 2 H's and an O are removed, 1 O is left behind and serves as a linkage between the 2 alpha glucoses. Because we're linking the alpha form of glucose, we call it an alpha linkage. Because we are linking the first carbon of one alpha glucose to the 4th carbon of another alpha glucose, we call it an alpha 1,4 linkage. And because we're linking glucose, we call this a glycosidic linkage. So again, as I've advocated for you before, the idea here is to learn the vocabulary and sound kind of fancy so that you can hang out with fancy science people and understand what they're saying and converse with them, just like if you were learning a foreign language. So we call this an alpha 1,4 glycosidic linkage, which is formed between 2 alpha glucoses via dehydration synthesis. A very fancy way of talking about this process that hopefully is now understandable to you all. Note I'm showing below, you can also break apart alpha glucose, and if you were to eat some starch, because remember that starch is what you get if you have a chain of alpha glucoses that are linked by alpha 1,4 glycosidic linkages, you make starch, also chemically referred to as amylose. You can break down starch in your gut, and so if you eat a starchy food for breakfast, lunch, or dinner, or perhaps all three, you will take the starch, the amylose, the alpha glucose chains, they will go into your gut, and water in your gut will be used to break the alpha 1,4 linkages by reinserting the HHO that was originally removed by dehydration synthesis. This is how you break your food down. So you break down starch in this way in order to turn it into individual glucoses, which you then absorb into your bloodstream. And once they are absorbed into your bloodstream, they can be transported throughout your body. And we've already begun to talk about that during Unit 2. How do they get across the cell membrane, for example? We're starting to look at that. So hopefully this is all feeling coherent to you all. You also know that if you flip the first OH group, and so I'll draw that over on the side of the slide here, and I'll clear my drawing so it's easier to look at. If I draw the other possible form of glucose, again, I draw a hexagon with an O in the top right. Here's my first carbon, my second carbon, my third carbon, my fourth carbon, my fifth carbon, and then I know glucose is C6, so here's my sixth carbon. C6H12O6 is glucose. So I put a CH2OH coming off of the fifth carbon. And then I go down, down, up, down if it's going to be alpha. But in this case, if it's the beta form, I'm going up, down, up, down. Many of you ask, why do we write the one on the left with sort of an HO instead of an OH? And remember, it's just to show that the O is what is bonding to the carbon ring of glucose. It's not the H that's bonding to it. So we clarify that the O is bonding to both the H and to the carbon ring. So now when we put two beta glucoses alongside each other, we see very quickly what the issue is. Oops. Up, down, up, down. Apologies for my sloppy drawing with a trackpad here. Up, down, up, and then down. And whereas alpha glucose lines up perfectly to link the 1 and 4 OHs together, beta glucose does not. The first OH in beta is up, whereas the fourth OH is still down, and they do not align, which means that the beta glucose actually needs to flip over so that this OH in the fourth position is facing upward and it can link. That flipping is the basis for beta glucose forming a different type of molecule when it is linked by beta 1,4 glycosidic linkages. Beta 1,4 glycosidic linkages makes a chain of beta glucoses, which have a different shape because of the flipping of every other beta glucose so that they can align properly, leading to a different polymer, a different chain called cellulose. Cellulose, as you learned, is the primary component in wood. That and lignin is the other molecule you'd find in wood. Cellulose is a reason why the toothpicks that we played with water with, that's why the water will stick to the toothpick, is because the toothpick is made of cellulose, and cellulose has OHs all over it. And as you know, when O and H share electrons, O wins in the tug of war and becomes partially negative, while H becomes partially positive. And those partial charges that are all over the cellulose allow the toothpick to cling to water a little bit. Water has partial positives and partial negatives as well. Okay. You do not have an enzyme in your gut for breaking beta 1,4 linkages. So interestingly, if you were to eat cellulose, we simply refer to that as fiber in your diet. Now, wood is technically a source of fiber, though I wouldn't recommend it. Get it? Wouldn't. Anyway, wood, not a good source of fiber, but if you eat any other stemmy plant material, or just one that is very leafy, very kind of plant mattery, it'll be high in fiber because plants make their structures out of cellulose. They make their cell walls out of cellulose. If you eat celery, the strings of fiber that you would see in a celery stalk, which is a stem, those are rich in cellulose. So lots of fiber in that. That isn't digestible, so it just passes right through you, right? Okay, so nice little indication there as we begin to understand the ways in which molecular structures determine the functions that emerge. And one thing that I'm hoping you're getting from this course is that it doesn't take radical changes in molecular structure to give very different properties on a larger scale. So there you have the positioning of just the first OH group on the glucose ring being either up, as in beta glucose, or down, as in alpha glucose, leading to a totally different style of polymer or chain of them when you link them together, which leads to extremely different properties. Either a starchy glucose storage system that an animal like me could easily break down in my gut and use as food, or a stiff, strong chain of cellulose fibers that can be used to like build something so strong and sturdy as wood or a vegetative plant stem like you'd find on celery, and which can't be broken down by most organisms. So structure determines function. Structure determines function. And that's a great example of how small molecular shifts can lead to very different functions, which is critical to understanding biochemical systems. Now, speaking of the molecules that make up biochemical systems, if carbohydrates are the product of photosynthesis, which indeed they are, as we've just seen, we should also note that there are three other major types of biological molecules that are used to make up living systems. Those three other types are listed here. Lipids, which are primarily carbon and hydrogen. We could also call them primarily hydrocarbons, is another way to talk about that. Proteins, which contain all of the elements of carbohydrates, C, H, and O. Again, as I mentioned to you, carbohydrates have the generic formula of CH2O, which is why they are called carbohydrates, right? Proteins have all of the atoms that carbohydrates have, C, H, and O, and they also have nitrogen. And nucleic acids, which include DNA and my very favorite molecule, RNA, which I love, those contain not only CH and O, as well as N, but also phosphorus as well. Now, the connection that we made in class is, since plants can make carbohydrates from photosynthesis, all they have to do is subtract the oxygen from their carbohydrates, and basically they have little carbon and hydrogen chunks that can be turned into the building blocks of lipids. So, if an olive plant does photosynthesis, it's going to subtract a lot of the oxygen from some of the glucose that it makes, and it's going to make oil, and we will call that olive oil, right? And that would be a lipid. It's basically just a chain of carbons and hydrogens. Now, on the other hand, if the plant is going to produce protein, it needs a source of fertilizer. And fertilizer contains compounds like potentially ammonia, NH3. Now, if I draw ammonia, you might be beginning to notice that these molecules are like actually all quite similar. Here's ammonia, and then here's the nitrogen-containing section of a protein. This is called an amine group. Has the formula NH2. So you can see, this is why a plant would want ammonia, NH3, because it could take one of the H's off and just stick that right onto some carbon and oxygen that it got from a carbohydrate, and it could build a protein, basically. And that's what a plant will do if you give it fertilizer. It's also hard to come by fertilizer, which is why plants are not famously rich in protein usually, and the ones that are the ones that are really good at getting nitrogen. Okay, we'll talk more about that later in the year, why certain plants are particularly good at getting nitrogen. So I know we previewed it in class a little bit. Phosphorus in the form of phosphate is the other, phosphate has a formula PO4, so I've circled it there on the nucleic acid, PO4 with a 3- charge. That's another common component of fertilizer. And again, you could see how a plant would take ammonia and phosphate and some carbohydrate that it made from photosynthesis and basically link them together to make the building blocks of DNA and RNA. So what I want you to realize is basically like living systems are the product of photosynthesis, or they are the product of photosynthesis with a couple of elemental modifications to it, subtracting oxygen to make a lipid, keeping the oxygen and adding in some nitrogen to make a protein, or keeping the carbohydrate and adding phosphate and nitrogen in order to make a nucleic acid. Okay, this is basically all the chemistry that you need to know to understand a biological system very in depth. It's all the same principles over and over again. They just build on each other and get a little more complex, but this is the foundation. Okay. Finally, right, just kind of going through these categories, I'll note for you, and this is not something I emphasized in class, but this is something that is going to preview things that are coming up in the next unit. The molecules I just told you about become linked together in ways that make up the structures that cells are made of. So for example, in this top panel, we're seeing how you can take an amino acid, which you just saw one of those on the previous slide. You can link it together with other amino acids. Guess what reaction is used to link it? If you said dehydration synthesis, you're correct. And then those linked amino acids, which we call a polypeptide, can be made into a cellular structure. In this case, we're showing an intermediate filament, which is a part of the cytoskeleton of a cell, basically the structure in an animal cell. This is what gives your animal cells structure. So we can see how building atoms into molecules can then lead to those molecules being linked together and made into cells. And again, my logic in this course is from atoms to molecules, molecules to cells. Then we're going to go cells to organisms, organisms to ecosystems. You can see how a building block of DNA can be linked to make DNA, which can be complexed together with some protein to make a chromosome, which is the cellular structure that contains the genetics of a cell, okay, the genetic material of a cell. Similar here. Look, here's which glucose? Down, down, up, down. That's alpha glucose. When it's linked together to make alpha 1,4 linkages, it makes starch. And then starch can be stored, like potentially in a chloroplast where photosynthesis is taking place, which is a very logical place to store glucose, since photosynthesis is what makes it. Do you see how it's the same stuff over and over again? And then finally, on the bottom here, we can see how lipids can be put together to make structures like a fat molecule, or as you're learning in our next unit, if you stick a phosphate group to the top of a molecule with two lipids attached, you can make a phospholipid. Phospholipids, where the tails are these lipid chains, these fatty acid chains, and the head is a water-soluble phosphate. Okay, so that's kind of what we're doing now, looking how these link together to form building blocks of cellular structures. For example, phospholipids build the cell membrane. All right, and then a little more detail. Here is the phospholipid structure that we studied in class, just so you can see it. You'll note up at the top that negatively charged phosphate. The negative charge is there. It's not shown properly in this diagram. There also, as was noticed in class, should be a second H right here because of honk bonding rules. That carbon needs to have 4 bonds. So 2 little errors in this image, but now it's fixed. And this whole head unit is the water-soluble charged part. These tail sections that hang down, again, we would draw the head unit as a circle, the tails as these 2 long chains. That is fat soluble. And so we've seen how these assemble themselves into a cell membrane by making a bilayer like this that becomes the cell membrane like this. Inside the cell, the intracellular environment is water-based. The heads are water-soluble, so that's cool. Outside of the cell, the extracellular environment is also water-based. The heads are water-soluble, so that's cool. In the middle here, there's no water. That's good because that's where the fat-soluble